So, as I was already introduced, my name is Tobias Peherstorfer. I am a PhD student at VRVis in Vienna, Austria, and today I have the opportunity to tell you all about some multi-omics atlases and how to integrate multimodal data across different scales of resolution in those atlases. So let me quickly just summarize a few facts about like the VRVis, who we are. VRVis is a non-profit research institute focused on visual computing and located in the beautiful city of Vienna. Our group, the Biomedical Image Informatics Group, is headed by Katja Bühler, and currently we consist of 3 researchers and 3 software engineers. So, to the outline of my talk, I'm first gonna provide you with some definitions. What are we actually talking about? So what is omics data? What is a reference atlas? Then why should we even integrate data like this into a reference atlas? Next, how did we at VRVis build our multi-omics atlases? Then also, how can these atlases be used to explore and query data? And finally, I'm going to give you a short demonstration with one of our web-based tools, namely BrainTrawler. This is a reference atlas exploration tool based on brains and specifically for brains. But of course, the whole concept of omics data and reference atlases is applicable to almost any biological entity that you want to have. You can make atlases for organs and whole animals as well, and humans, of course. Okay, so let's start with the first definition. What is omics data? If you look up the definition of omics data, it is basically saying that omics quantifies biological entities in tissue. That is kind of abstract, so let's head into a specific example, the prominent field of transcriptomics. In transcriptomics, you try to quantify the amount of transcripts, so in that sense, RNA that is available in a specific tissue, as many of you know. RNA is a product in the process of making proteins from a gene, and therefore the abundance of some RNA in tissue can give you a hint at the activity and the relevance of the gene in a specific tissue location. There's also other omics data types like proteomics, which quantifies the abundance of proteins in tissue. But there's also omics types that actually don't concern themselves with, let's say, molecules per se, but with other biological entities like connections. These are, for example, even, let's say, these are some of the most important for neuroscience in general, and they are summarized under the umbrella of connectomics. In connectomics, we concern ourselves with either structural connections, these could be neurons, could be on a larger scale axon tracts and nerve bundles, but they could also be functional connectivity like you would obtain from resting state fMRI. Of course, there's also tons of other omics data. There's genomics, epigenomics, metabolomics, and they all differ by basically what exact biological entity they try to quantify. Now, as these are the data days and we want to talk about this kind of data with the lens of a data scientist, for us at VRVis, it is always important when we look at such data to ask at which resolution does this data come and how is it anchored in space? There are 3 main options for this. First off, we have what we call dense data. Dense data would commonly be acquired through imaging and is characterized by the fact that there is data available for each pixel or voxel in the image. So, for example, in MRI scan, microscopy images, you get it. On contrast, there is what we call sparse data. Sparse data is characterized by the fact that we only have data for specific points in space, and this data is usually acquired in methods that use tissue sampling. We'll get to specific examples a little later. And finally, there is the data type that we call semantic data. This data is stuff you usually get from literature, where there is not a specific location in space reported, but rather a construct like a brain region, a part of the lung, some standardized construct that it is linked to. So there's only a semantic link to space. Now, To really understand these concepts of resolution that we talk about here, let's head into a few more details of examples for omics data. I'm just gonna list like a common method, tell you which type of omics it is. I'm sure all of you will know, and then talk about which resolution we will classify that in. So first of all, I'm gonna mention microarray. This is a method for measuring gene expression, and it is usually tissue sample based. So we will most often get sparse data, but also I'm always writing semantic here as well, because you could also obtain the data from literature. Next, we could investigate something like 10x Visium. This is a spatial transcriptomics platform, which is imaging based and gives us transcriptomics information over a whole slice image. So we get dense data from it. There would also be techniques like single cell RNA sequencing. Sequencing methods, of course, there's spatial transcriptomics, which can give us sequencing methods that are more highly resolved, but like standard sequencing pipelines often use tissue sampling, which is why this data is also considered sparse by us. Let's head to another field of omics. Let's head to connectomics. I've mentioned before, you can get connectomics data, functional connectomics data, by, for example, using resting state fMRI. And since fMRI is an imaging method, we get dense data from it. There is also, on a very much higher resolution level, the possibility of getting electron microscopy images and then tracing neurons there. This data is also dense, since we rely on imaging data to get it. And finally, let's get to another omics type. This one is epigenomics. ATAC sequencing measures the accessibility of the chromatin and is usually also based on tissue samples. So again, we say it's more on the semantic or sparse side of resolution. And as you can see, all these very different types of omics, of methods of acquiring omics data boil down to exactly these three types of spatial resolution. Okay, now let's head on. What is a reference atlas? Definition number 2. So a reference atlas is basically a standardized reference framework for an organ or an animal or anything you want to align your data to, that provides you anatomical context and lets you compare this data to each other. So it consists of 4 main ingredients. The first of which is a reference image. Again, I'm going to stress I'm using brains here because we are working a lot with brains, but you could also be using lungs or the kidney or any organ you like. Usually these reference images are either taken from literature, like medical books, or produced from high resolution slice images from microscopy, or even from averages over MRI images with very high resolution. You pair these reference images with the coordinate space they live in to be able to assign each point in this reference image a specific coordinate. That makes it easier to then connect all your data. And then we want to add a sense of context in this reference image. We want to subdivide it into smaller portions. And for this, we use what's called a parcellation. Now, a parcellation is just a way how we divide up our reference image, and it can be based on multiple different merits. It can be based on anatomy, on function. It can even be based on the most prominent cell type in that type of tissue that you group. And the final ingredient is what we call an ontology. An ontology is a standardized way of naming and organizing your parcels from the parcellation so that you can reference them outside, and you can know if they are, for example, hierarchical, like many brain regions are. You can expand and de-expand them and investigate child and parent relationships. Okay, now next off, why now have I told you this? What is the use of such a reference atlas, and why should we integrate our data into a reference atlas instead of just analyzing it as it is? So integrating data to a reference atlas means you align it with a standardized and probably widely used reference image and reference space, which means it promotes data standards. It promotes that your images are fitting and probably also registered to that image, to that reference space. It also promotes that these data that you integrated with the reference atlases can be reused because many people are likely to use the same or know the same reference images. They promote data democratization because often reference atlases and multimodal atlases can be accessed publicly or are made publicly. And finally, it's also great for education because you can easily provide students with the anatomical context needed to understand your data in a spatial domain. For us as data scientists, there is an additional benefit, and that is that for integrating data into a reference atlas, it basically provides the spatial and anatomical context. And that is what we're also going to use for the integration. Now, let's talk about how we built our brain atlases. First off, I'm going to tell you only about the reference atlases. Then I'm going to mention the data that we integrated, and then I'm going to tell you how we did that. So first of all, at VRVis, we have maintained 3 reference atlases with data ingested to them. First, we have a human atlas. This uses a template brain from the MNI Institute, specifically the MNI 152 nonlinear template. This is an averaged a template that was generated by averaging over 152 high-resolution MRI scans, and it also comes with a specific coordinate space. And we pair that up with the Allen Human Brain Reference Atlas, which is a parcellation that is already fitted to this specific template and an ontology in itself. So basically, this is all our ingredients that we need for the human reference space. For the mouse, it's even easier. The Allen Brain Institute actually provides a full reference atlas that you can just use as it is. It's called the Allen Mouse Brain Common Coordinate Framework. And it provides you with high resolution slice and 3D images and a detailed and fine-grained parcellation and ontology. And finally, we also have a reference atlas for the fruit fly, for the Drosophila larva. Together with collaborators, we have constructed a template image by averaging Gulf-4 staining images, and then also have defined a parcellation and ontology together with them. Okay, these are our 3 reference atlases that we're currently working on, or that we already have, basically. And the data that we have integrated is actually a lot, and it focuses largely on gene expression data. For the mouse, we have integrated the Allen Mouse Brain Atlas. So this is in situ hybridization data, so imaging data, along with a number of single-cell RNA sequencing data sets that together give us full brain coverage of cell-type specific gene expression data in the mouse. In the human, we integrated the Allen Human Brain Atlas because it is actually very, very well known and widely used, along with the combination of bulk and single-cell sequencing data sets that again give us full brain coverage in the human. And for the Drosophila larva, we have 3D Gulf-4 staining images that highlight specific neurons and even annotated 2D Gulf-4 images, staining images. Both of these are for the L3 larva stage, so for not the first larva stage, but the third. And yeah, so that is also quite cool connection. Maybe important here, the human and mouse data is already published and publicly available, and the data for the fruit fly is not yet publicly accessible, but will probably be until the end of the year. Connectivity information we have integrated is for the mouse brain, we have integrated the Allen brain axonal projections data set that is basically viral tracing of neuron bundles in the mouse brain. And for the human, we have a resting state connectome from the Human Connectome project. We also plan to integrate CFOS imaging for the mouse for task-based for tasks and treatments, and for the human to integrate meta-analytic activation maps from Eurosynth and publish that also soon. So now that I've bragged a little about the data that we have, let me get into how we actually got it there. You all will remember that I told you before about these different resolution types with dense data, sparse, and semantic. Now let's look at each of these types and let's see how we can integrate these data. When you first get your dense data, it will not be aligned to your reference space, not to your reference image, sorry. And the first step you will need to do is register it to this image and then resample it. I mean, I'm oversimplifying. All these steps are quite complicated, but this is what you need to do. And then to make the multi-omics atlas that you're trying to build really useful, you're also going to have to index your data. This is a step that is quite important for being able to search and query your data later. But I'm not going to talk about it too much now. Let's head on to sparse data. As you remember, sparse data, in contrast to dense, we only have specific points in space where the data is available. And there is actually two main ways of converting this kind of data to be integratable. And both of them actually aim to make it into dense data. So you can either aggregate your sparse data in parcellation regions, or you can just splat the data in 3D and assign volumes around these data points with the values you have from the sparse data. Both of these methods turn this data into dense data. Then you can proceed as normal as you would with dense data for integrating and finally also get to the indexing step. Now, in the last step here, we have semantic data. And as you can see, there is a fork in the path. And this fork basically depends on the answer to the question, is there a mapping to 3D of the semantic data? What do I mean here? The semantic data lives in an ontology. For example, here, the Brodmann areas of the brain. And if we know how this Brodmann area maps to 3D in our reference space, we can leverage this. But let's pretend we don't. So we don't know how it maps to 3D. Then we need to manually identify regions that correspond to each other in the ontology of that the data comes in versus the ontology that we actually have in our reference atlas. So we need to do this mapping manually and then assign regions with this data. So this is a cumbersome process. It is easier if you really have the 3D mapping, then we can just use the voxels that we know Brodmann area 4 corresponds to and say, okay, we paint those with the values that we have. And finally, we go to indexing. So now I've dodged the topic of indexing long enough. Let me get to it. Indexing, as I talked to you before, is basically about enabling queries fast. So we want to be able to explore this data really fast. And the keyword here is spatial queries. Spatial queries are the process of, let's say, asking in your reference space, you pick a specific voxel and you ask, what are the values, which genes are highly expressed here? You ask that question to a, let's say, in situ hybridization dataset. For each gene, you have a 3D image that gives you the values of that gene expression in space. So you would have to go through, let's say, 20,000 images, identify, hop to that point in space in the memory, and then assess it. This is quite inconvenient and not very efficient. So there is a smarter way of organizing your data on memory to make this work faster. And the way to do it is actually organizing your data to be not stored per image, but per voxel. So you store all data of a single voxel of all images in the same, let's say, location in storage, because then you can just say, okay, all data from that point is here in that specific memory location, and that allows you to really quickly get to your data. But there's another problem here. What if I want to have, let's say, a selection of 4 pixels that are close to each other? This again would open up the problem of having to jump around in memory and looking for stuff which is not so efficient. So additionally to ordering our memory in this weird and unconventional way, we can also use what's called a space filling curve. A space filling curve is basically a curve that meanders through our reference space and lets us assign each voxel to a specific position on the curve. And usually, if you would say we want to find all voxels that are in these yellow marked regions, you would need to go from left to right through the entire file and say, okay, we find the first one here, the second one here, jump again, left to right, and then you're on the second row and find the next one, the next two ones. But with the space filling curve, all these, if you order your data along this curve, or usually your data is much closer together in memory if you make a spatial query in in close proximity to each other. So that is really a way of how you can speed up your queries even more. Now, this is good for volumetric data. For network data, it gets a little more complex, but you can also do this. So network data would be connectomics data, because of course, this is not really imaging data in a sense that we have, for example, for fMRI, but this is more a network, as we would call it as computer scientists. And there you can do similar stuff. On the upper left here, you can see a heat map of a connectivity matrix that is not ordered. It's a voxel level resolution. That means for each voxel in our image, we have one row and one column, and we see the connectivity between those. And by just reordering it along such a space filling curve, we get to the second stage here, which already shows us that actually, okay, it's not that bad. Apparently spatial proximity is kept quite well when we reorder like this, because now it looks much more homogeneous. And then you can do some clever shenanigans by compressing rows here and already pre-computing cache files to make connectivity, so queries on such network data really, really fast. Important fact to know here is these matrices are usually like on the verge of like 20 gigabytes large. So searching this is not easy. So it's actually quite cool what collaborators of mine have done here. I refer to you to this publication that's listed here for more details. Now, since we have now explained this indexing stuff also, it's good to know what it can be useful for. Since with this indexing, we actually have real-time access to all the values of all images we have integrated. For any arbitrary selection we make, we can ask our application, hey, in this brain region that I'm showing you right now, where is this connected to? And it can easily answer the question and fast as well. We can ask for any arbitrary drawing that we make in our reference atlas, which genes are highly expressed here. If we have transcriptomics data integrated, we will be able to answer this question quite fast. And also we can even create selections from other omics data. Let's say we load up a task fMRI activation signal, we threshold that, and we take all the voxels that are with high activity and take those as selections, and we can then use these selections to query other data. So we can even make the connection between different omic data types via these spatial queries. And that is really the beauty of a reference atlas, because you can use space to connect different types of data here. By combining this also with some visual analytics methods, you can actually ask some quite detailed questions to your data, which is what I'm going to show you next. I didn't check the chat, by the way. Is there any question, or are we good?
No, so far we were good, but
Okay.
I'll let you know if we get anything in the chat.
Okay, cool. So yeah, then now it's important also to consider, now that we've done all this complicated stuff by integrating the data and making it queryable fast, how can it actually be used? How can it be accessed? Now, for these multi-omics atlases, there is usually two main ways of accessing them. The most easy and user-friendly is probably a web-based access, where there is a graphical user interface and very flexible general query options that are there to allow people to ask very general questions. You combine this usually with interactive visualization so that people understand what they see. And this is quite useful for education, as I also mentioned before, because here you really have a lot of spatial and anatomical or functional, depends on your postulation, of course, context for your data. On the right-hand side, for example, here you can see the Zebra Explorer, which is a eBrains tool that allows you to explore the multi-level human atlas of eBrains in your browser. It's really cool. It looks amazing. And yeah, I think it's a really cool tool for education as well. The other type of access for such multi-omics data is actually public APIs, where you can call different handlers of this atlas and get data directly in scripts. That makes it easy to integrate into self-built workflows and is very reproducible because you just write down what you did. You don't need to remember, you've already written it down. You can use the script anytime you want. So these are the two main ways such data is then accessible. Okay, that was it with the first part of the presentation. Now I'm coming to the demo. I'm going to show you the BrainTroller application, which is focusing on exploring and visualizing data for the brain, multimodal omics data for the brain. And this version that I'm showing you now does not yet contain neuroimaging data, like I promised before, and also not brain circuits, but this will come soon in the future. Okay, let's head there. Yeah, okay, no, we don't need to check this. So, this is a landing page. In BrainTroller, you can choose between exploring data for the mouse and the human brain. Let's head to the human brain first. That is bad. Let me quickly check what's happening here. 1 second. Okay. Yeah, this should be better. Okay. So now it works. So BrainTroller is designed or was designed mainly with the focus on network data and transcriptomics data. So connectomics and transcriptomics, these types of omics data. There's the first page here, the light page, immediately gives you a spatial overview of our transcriptomics data that we have ingested. What you see on this heat map here is on the y-axis, we have all our data sets that we have integrated. They are, if there's a commonly used name for them, we use that. Otherwise, we use the first author of the publication plus the publication name here. And on the x-axis, you see all the brain regions in our parcellation. And the tiles in this heat map tell you how many samples we have for each combination of data set and brain region. So this is important because transcriptomics data sets, especially single cell RNA sequencing data sets, usually don't cover the entire brain. Of course, there are newer, more exciting data sets that almost do, but with the collection we have here, there's a lot of data sets that cover specific brain regions only, and this page already can give you an overview of where to look for. If you're interested in data for the frontal lobe, you know that there's a lot to choose from. But if you're interested in the midbrain tegmentum, you need to really look for these specific data sets. The cool thing is, since we have integrated our data with a hierarchical parcellation that is organized hierarchically, we can even say, okay, yeah, I'm interested in the frontal lobe, but actually I'm interested in a subregion. Let's look at a subregion of the frontal lobe. We can do that by just opening the parcellation that I've showed you before. This is the ontology that organizes our brain split up, so to say. And we can just navigate to the frontal lobe here and say, okay, let's zoom in a little. Let's expand all the subregions here. And indeed, this plot already updates and shows you the exact distribution of samples among all our subregions of the frontal cortex. Okay, so let's explore a little what we can do here in BrainTroller. I'm gonna go to network query here. This page is meant for querying connectomics data, and you can select a connectivity matrix like the resting state connectome here, and also a spatial ROI. This means that we basically ask where, let's pick the thalamus, not the fourth ventricle, thalamus. So we selected the thalamus in 3D here, and in 2D you can see it. So here it is. And now we can ask for this specific connectivity matrix, what are the incoming and outgoing connections of the thalamus? Of course, this is resting state connectivity, so ingoing and outgoing is the same, it's symmetric. But anyways, this is relevant for structural connectivity where we have directions. And we can ask, okay, what is the outgoing connections of the thalamus? We can easily click here, and in a few seconds we get both a visualization in 3D, 2D, and a region profile that shows us exactly where the thalamus is connected to. Now, in this image, you see that it's connected apparently widely in the brain. You get more detailed information in this profile right here. Each bar here stands for a specific brain region of our parcellation. And you see the thalamus apparently is connected very well to the thalamus, which is good, of course. But it's also connected to a few other regions. For example, the transverse temporal gyrus. Let's expand that by double clicking. Again, this opens up this specific region in the parcellation that we also used before. And let's also pick some other region that apparently the thalamus is highly connected to. Well, let's pick this here. And what we can now do is we can say, okay, we've searched for regions the thalamus is connected to. We can now pick those regions like this. We have now set them as spatial selections. And now after picking these regions, we can navigate to gene expression query and then use our selection or informed selection from the network query and from the network data that we saw to query another data set that is transcriptomics based. For example, we could query the Allen Human Brain Atlas. It already tells us that the Herschel's gyrus is covered by 28%, is covered in 28% of its entire volume by this data set. And same for the other regions that we have selected. And we can run different kinds of expression queries here. So the standard expression query, which is what we're gonna run, just asks for the average expression of all genes in the marked region. So we go through all the voxels in here, count up the expression values, and then rank order our genes by that expression. And indeed, we get a result list here that shows us the gene name, the full name, entrée ID, ensemble ID, so the standardized IDs for each gene, and they are ordered by the expression value here, rank ordered, and you can even investigate the gene expression in 3D. Of course, this gene is quite widely expressed everywhere, so it just glows greenly. Okay, now the cool thing is you can even, as I said before, combine queries to ask more detailed questions. We now ask the question, which genes are highly expressed in regions functionally connected to the thalamus? Now let's compare that with regions with genes that are highly expressed in the thalamus itself. We can also ask that question. So let's pick the thalamus as our region of interest, and let's execute also another expression query. We will get another table down here. It's just loading, and here it is. Now, it would be cool to see the difference between those tables, right? To see the difference in the connected region to the thalamus and the thalamus itself in gene expression. Of course, it's kind of cumbersome to go back and forth to compare these tables, but there we have actually a quite cool page to do this. In the gene expression analysis page, we can use what's called a parallel coordinate plot to easily compare these two lists. So in this plot, the first y-axis here is our first search. So the list of genes that are expressed highly in the connected regions to the thalamus. And on the right here, we have genes expressed highly in the thalamus. We can brush just by clicking and dragging a portion of this region. Each blue line that connects here is one gene. And we can just say, okay, we want to find genes that are highly expressed in the connected regions of the thalamus, but not so highly in the thalamus itself. Indeed, there are two genes. There are only two genes that fit the criteria, and we can easily investigate them here. We can see the expression in the thalamus and in the connected regions here. And by combining all these tools, you can really hone in and ask very detailed and complex questions to your data sets. And all of this is actually only possible because we have integrated our data with a reference atlas. So we know what a thalamus is. We know that how to get to the connected regions. We know how we can really use space to interlink these different kind of omics data here. Okay, so that concludes my demo. At last, I want to thank my team also for the support, and I'd like to thank you for your attention. You're welcome to visit us and also use BrainTroller. It's publicly available, free of charge. You don't need to log in. You don't need to upload any data of your own. You can just use it. Yeah, and that's it. Thank you.